The overall goal of the following experiment is to assay the pathogenicity of clinically significant non-synonymous changes using in vivo complementation in zebrafish. This is achieved by first generating mutant human mRNA to recapitulate human genetic lesions. Next, human wild-type and mutant mRNA and morpholinos are injected into zebrafish embryos and translated into functional protein or used to block translation, respectively. Then, the embryos are phenotyped in order to evaluate the effects of the mutant protein on development. Results are obtained that show the deleterious effects of mutations on human protein sequences that can be confirmed based on the rescue capability of mutant human mRNA.
The main advantage of this technique over existing methods, like the mouse model, is that potentially damaging alleles can be more rapidly assayed at a medium to high throughput level. And also, allelic series within a single gene can be evaluated to better understand the direction of effect at a cellular level, such as loss of function or gain of function.
To begin, determine if the human gene of interest has a zebrafish ortholog, and if so, how many copies. We recommend reciprocal blast of the human protein against the zebrafish genome and subsequent blast of the best zebrafish hit against the human genome. True orthologs will be the best hit in each instance. Obtain or generate a construct using the human open reading frame for the desired gene less than 6 kilobases in length, as well as a 5' sp6 transcription site and a 3' poly A signal.